peels away this bottom strand. Okay, start moving to get to TL. Okay, so remember when you cut, you have a 5' and a 3' hydroxyl group. And who is going to recognize this 3' hydroxyl group? DNA polymerase. So DNA polymerase, you cannot have this stuff without being single-stranded. It's going to immediately start filling this, copying the top strand, and at the same time helping to peel away this strand that is attached via the complex with these other proteins. Okay, so you have DNA synthesis. Then when we get to this point, TL, this is cut, this strand is cut, and the other proteins fall off earlier once the cut is made, and you have a single-stranded tDNA attached to VRD2. Okay? This is the molecule that is going to be transferred to the plant cells. So the protein coded by the VRD operon, except VRD4 and VRC operons, are involved in generation of the tDNA strand, and VRD2 binds to a specific site on the RB or TR of the tDNA. It next, the bottom strand binds covalently to this nick DNA at the 5' end in a transesterification reaction, which is analogous to that carried out by relaxase. You guys need to keep up with the material. It's going to get denser and denser. And I'm not going to be doing quiz after quiz after quiz to give you extra possible. Really need to study. So VRD2 is analogous to relaxase. So VRD2 peels away this tDNA strand, and as this is happening, as you saw in the diagram, then DNA polymerase is initiating synthesis in the opposite direction to restore the double-stranded DNA. You cannot have gaps of single-stranded DNA because they will be degraded. So the rest of the VRC and VRD proteins fall off the complex. VRD2 remains attached all the way till the transfer into the plant nucleus. So, okay, so as I mentioned, the tDNA is cut at the left border, TR, left border or TL, sorry, and then we generate it, as I showed in the diagram, single-stranded tDNA molecule with VRD2 attached covalently to it. There is a phosphodiester bond here, joining the DNA to a particular tyrosine on this protein. Okay, so next time I'm going to then describe what is the transfer, how is the transfer apparatus, who makes what proteins make that, and other stuff to finish this story. So I will post this tomorrow, not today, because I have a long meeting this afternoon and I teach tonight. But keep up with the material as I go along. Are there any questions?
Yeah, Dr. Benuit, you said that the RB was the same as the TR, right?
Yes. Okay. Yes, they are exactly the same.
For the quiz, you'll post some information beforehand, right?
Yeah, but not today. Today I am completely, my time, I cannot do it.
All
it's
right.
not
Whenever.
possible.
Yeah.
Physically
Yeah.
not possible. Yes. Okay.